Hello, everyone. Welcome to one more BrainMap seminar. Today, we are very pleased to have Professor Ivan Luptak. He's MD, PhD. He's Associate Professor of Medicine at Boston University School of Medicine and a cardiologist at Boston Medical Center. His research focuses on cardiac energy metabolism, mitochondrial biology, and translational imaging approaches to hypertrophic cardiomyopathy and metabolic heart disease. His laboratory employs multinuclear MRI spectroscopy, as we are going to hear today, in ex vivo Langendorf perfused hearts, in vivo phosphorus NMR, and multi-omics analysis to define energetic dysfunctions and test novel interventions. As always, this seminar series is co-sponsored by P41 Funded Center for Mesoscale Mapping, housed here at Martinos Center. Thank you so much.
Thank you, Karen. All right, so let's dive right in. All right, so as cardiologists, we are always interested in the heart's pumping function, the mechanical work. But in order for the heart to perform this work, it needs to function as a energy converter and convert energy, chemical energy from nutrients to mechanical work while creating also heat and entropy. And this all is mediated by ATP production in the mitochondria, which we'll talk about later a little bit more. So in this talk, I'm going to tell you first a little bit of overview of my understanding of the heart as a thermal engine. Then we spend some time on phosphorus NMR, C13 NMR, and a little bit on sodium NMR. And throughout, I'm going to be mentioning some applications and publications we had on this in terms of metabolic interventions to fight heart disease. All right, so conversion of energy in the heart happens first in mitochondria from nutrients and oxygen, where mitochondria basically burn in controlled manner the chemicals, the nutrients, and create ATP. Then ATP has to travel to the place where it's needed to perform work, such as sarcomere or for pumping function of the ions. And both of these processes have around 65% efficiency. So all in all, it's about 4% efficient process. And so how do we look at this? So with C13 NMR, we can look at the contribution of different substrates to oxidative phosphorylation and ATP synthesis. And with phosphorus, we can then look at how much ATP fluxes are there, what is the energy charge of the heart. So heart is an omnivore, but in terms of development, when the individual is first born in fetal development, the heart is relying mostly on glucose for energy production, and then adult heart switches towards preference to fatty acid as energy source. Then there are some situations where in fasting or type 1 diabetes, this can be even more dominant, the fatty acid contribution. But mostly in most of the diseases, cardiac diseases, there is actually switch back to carbohydrates as a predominant source for energy production, such as ischemia, hypertrophy, and heart failure. So the models we use in my lab to mimic the cardiac disease are two major models. Western diet, that we feed mice with high-fat, high-sucrose diet. They become obese, develop insulin resistance, and cardiac phenotype characterized by left ventricular hypertrophy and diastolic dysfunction. The second one is HCM, which is a mutation in sarcomere causing hypertrophy of the heart. And the mutations we use usually mimic the human disease and to develop similar phenotype, again, characterized by thick hearts and diastolic dysfunction, but also lack of contractile reserve, which I'll mention later. So, and in all this, there is, as I mentioned, the switch to glucose as a predominant source, carbohydrates as predominant source of for ATP synthesis. And we then are searching for interventions which could push this back to the profile, normal profile for adult heart with predominant fatty acid oxidation. So what are these interventions? One of them is SGLT2 inhibition. These drugs have been developed originally for treatment of diabetes, but approximately 10 years ago, it was found out in a major trial published in New England Journal that cardiovascular outcomes were decreased in patients treated with SGLT2 inhibitors. And since then, there is a boom of approaches with SGLT2 in heart failure with systolic dysfunction, with diastolic dysfunction, and improving outcomes across the board. So that's one way how to do this. Then we also have an approach, which I'll show you, where we can perfuse heart with a short chain fatty acid, butyrate, that actually bypasses all the transport systems and goes directly to mitochondria, feeding beta oxidation there and improving energetics. Then another one is kind of more interesting, maybe, where we increase the skeletal muscle mass of the mice, either genetically or with drugs, and that switches the metabolism in the heart in interesting ways also to predominant fatty acid oxidation. And I mentioned last one, incretins, because that's the one which I had collaboration here with Martinos Center. And the question is still there if this truly switches the metabolism the way we would expect. So one of the SGLT2 inhibitors which is used in clinical practice is empagliflozin. We had a recent paper on this showing that in HCM, in hypertrophic myopathy, cardiomyopathy, this not only improved metabolic efficiency, but actually reversed LV hypertrophy. Then in terms of skeletal muscle, this is early work. We only have abstracts on this so far, but improving skeletal muscle, increasing skeletal muscle mass did improve diastolic function and hypertrophy in metabolic heart disease. The idea here, just to introduce this, so you have this in your mind as we go through this, is that the insulin resistance is caused by increased intake of fat and sucrose in the diet, mimicking the Western diet. And if there is not enough muscle, maybe I can use my pointer. Yeah. So if the muscle is small, there is not enough diversion of the substrate there. But the moment you increase the muscle mass, it acts as a sink for glucose and branched-chain amino acids, and thus relieving the heart and other organs in the body from the overload of the nutrients. And the hypothesis is that it would improve energetics, it would decrease activation of the mTOR pathway, which is responsible for LV hypertrophy, and normalize systolic and diastolic function. And butyrate, that's one paper we have. First author there was Marcello Panaggia, whom some of you may know because he worked here at Martinos. He's a cardiologist and now at Novartis. And here we perfuse the heart with butyrate, which improves energetics and contractile function in metabolic heart disease as well. I will come back to this a little bit more later. Incretins, and here I want to show Karen's work, where we did hyperpolarized C13 NMR on hearts with systolic heart failure caused by a myocardial infarction, and then used hyperpolarized pyruvate to look for the switches in metabolism there. It's a complicated study because we had 3 interventions, different combinations of incretins, which, as you may know, Ozempic kind of drugs, which decrease the weight, cause the weight loss in these animals, and improved function and improved exercise tolerance, and also was changing metabolism in somewhat unexpected ways. I'm not going to spend time on hyperpolarized NMR in this talk because there's something which everything I know about it is from people in the audience or in this institution, so I'm not going to talk about that. I'm going to talk about the C13 non-polarized NMR. So again, when we want to look at energetics, we use 31 phosphorus NMR to better understand the transfer of energy between the ATP and the place where it needs to be used. For this, we have a phosphocreatine system in the heart. ATP is a large molecule which wouldn't easily diffuse to the place where it's needed. So there is the system of phosphocreatine, creatine kinase system where creatine takes on the high energy phosphate and brings it to the place. It's much smaller molecule, so it can go there, and then releases the phosphocreatine to ADP, regenerates ATP, and ATP then can power the contraction. The same may happen with ion pumps such as sodium potassium pump. With phosphorus NMR, we have a ways to look at ATP concentrations, phosphocreatine concentrations, also at the fluxes in this system. Another way how we can modulate the system and trying to optimize the efficiency of this thermal engine is when we try to not change the energy transfer, production or transfer, but when we influence the sarcomeric function. And we use small molecules called myotropes. These are small molecules which activate myosin ATPase and increase contraction directly at the sarcomeric level. This is the idea here is to use this in dilated cardiomyopathy, heart failure with systolic dysfunction, low contractility. These drugs are in development. They're supposed to be better than inotropes. Inotropes are medications which do the same, but through CAMP signaling, beta adrenergic receptor and CAMP signaling, increasing calcium cycling. These drugs actually do not change the calcium cycling. They only increase the energy consumption at the place of use, which is the myosin ATPase. And myosin inhibitors, why would we need myosin inhibitors? Well, we do because HCM, the sarcomeric mutations in So in terms of the activation, the myotropes, we had this molecule called troponin activator, which increased cardiac contractile function. And we compared this to the butamine, the classical inotrope. It had no negative impact on energetics, as opposed to the butamine, which actually depleted energetics throughout the experiment. And in terms of myosin inhibitors, again, this is used in hypertrophic cardiomyopathy. The big drug there is mavacamten, which is clinically widely used now. And we use different one called aficamten in a model of HCM, where it improved energetics, reduced hypertrophy, and improved diastolic function. So we have all these cool ways of modulating the system and trying to make the thermal engine more efficient. Importantly, also, I mentioned to you that ATP obviously is used for other things in the heart, not just contraction, but pumping ions as well. One of the important pumps is an ATPase. If that's impaired, it's unable to maintain low level of sodium in the cytosol. The moment you have too much sodium in the cytosol, you impair efficiency. And one of the ways you do that is it should deplete mitochondrial calcium through the exchange in the mitochondria called NCX mito. And then mitochondria, if they don't have enough calcium in it, they become inefficient, producing less ATP and more oxidative species, reactive oxidative species, and also resulting in contractile dysfunction. So the way to look at this is with sodium NMR and also rubidium. So this is just an example where we measured intracellular sodium with sodium NMR in isolated hearts. And SGLT2 inhibitor, this is a different one, this is ertugliflozin, actually decreased this elevated intracellular sodium and improved energetics and contractile function. And I'll mention a little bit more about it. So here, I just want to switch to phosphorus NMR, go a little bit through it, what we do with that. And I originally showed you the important thing here is ATP, and we do measure ATP concentration with sodium NMR. with phosphorus NMR. But what we really want to know is delta G of ATP hydrolysis. It's a thermodynamic complex thermodynamic term, which I'm going to try to explain my understanding of it, how it works. So free energy of ATP hydrolysis is something like this. So if you have a molecule and it falls apart, it releases energy, and that's your delta G, and it depends on change in enthalpy, which you can think of as a release of heat, right? If and also increase in entropy. If the entropy increases, which clearly does here, right? It falls apart. There is more parts, smaller parts, more of them. Entropy increased. So this would be a negative number, and you would derive a lot of energy from this process. You can also imagine there are already 100 other slides like this falling around. And in that situation, the changes wouldn't be as big because we almost know where to fall, and increase in entropy would not be as big. When we calculate this delta G in the heart, we first need to calculate ADP. So this is the thermodynamic description, and here is how we actually do it for ATP. To calculate ADP, you first need to measure ATP concentration and phosphocreatine. And we do this all the time. I'll show you the spectrum, and many of you know how phosphorus spectrum looks like. But you immediately get phosphocreatine and ATP concentrations there. What we also need is pH, which gives you the proton concentration. And we also need to measure creatine, total creatine biochemically from the extract from the heart, right? But already here you can see that if the phosphocreatine is high and ATP low, this would be a lower number, and thus ADP concentration would be low. So what you really want, you want a really high PCR to ATP ratio. That's like poor man's delta G. If you have high PCR compared to ATP, ADP will be low, and then when you plug the ADP here in this equation, this is the standard energy of ATP hydrolysis, with if all ATP, ADP are 1 millimolar and 100 micromolar of magnesium. These are constant. This is temperature. This is gas constant. And you can see here, again, you have ADP here divided by ATP. So the more ADP, the less negative number you will get for the delta G. There are some other analogies people like to just understand what it really means. If you have high delta G, it's like a full voltage in the battery, and you have some ATPase, such as this one, which requires certain voltage to work. And as you're approaching the delta G threshold, it still works, but there is less reserve. And when you go below the threshold, it stops working. And another one I also like is the waterfall analogy. You have ATP concentration, ADP concentration. If ADP is low, waterfall height is high. You derive a lot of energy from the ATP hydrolysis. If there is too much ADP, you can imagine it to be, as much as ATP, or you could reach thermodynamic equilibrium and derive no energy from ATP hydrolysis. So if there is a disease which increases ADP, you will have less delta G ATP derived. And it's important to note that the ATP concentration usually doesn't change. In many of the diseases, when we measure ATP is the same. What changes is PCR to ATP ratio, and that translates to lower delta G. So many of the processes which normally would work no longer works, or no longer works efficiently. And this is just to mention the important ATPases in the heart. Circa is the one which pumps calcium back to the SR during diastole. So the only way for heart to relax is that if Circa pumps the calcium back very quickly. So, and this is the most sensitive one. The range of for delta G is 55 to 65 negative delta G. This is around 52 threshold. This is the first one to go with ischemia or any kind of decrease in energetic status. Then NK-ATPase is next, and myosin ATPase actually goes last. But in ischemia, you usually get below that. So I'm a cardiologist, and so why should I be interested in all this? So this paper is almost 30 years old now. It was one of the seminal papers published where phosphocreatine to ATP ratio predicted mortality better than some other measures which we usually use in heart failure. This was dilated heart. So when we look at the New York Heart Association class, this is how much they can do in terms of exercise. And this one is ejection fraction. You can see that PCR to ATP ratio, the spread is much bigger and was actually better predictor of mortality than the other two. So that's why we're interested in this. So how do we do this in the heart? We perfuse them in Langendorf mode, which I'm going to show you. And what we can do there are kind of what I call basic measurements, because as you perfuse it, you have a balloon in the heart, you can measure systolic, diastolic function, contractile reserve. You can do ischemic perfusion, and you get the high energy phosphate concentrations. Those are basic measurements. You get phosphocreatine and ATP. And then you can calculate delta G, as I just told you. And then you can measure fluxes. And we'll talk about that a little bit more. So this is how it looks like. You take a mouse heart, put the balloon in. You can pace it. This is outside the magnet. This is easier. The balloon in. You can do pressure volume curves like this, increasing the balloon volume. You can see systolic. As you increase the volume, it's one of the things we can do. This is, how it looks like. It goes to the 10 millimeter tube, and the tube goes to the magnet. Obviously, in real life, it has to be connected somehow. So the way we do it, we call this umbilical cord. It's like a water jacketed hose full of tubings, which then go into the magnet and maintain the heart beating inside. This is your typical phosphorus spectrum with phosphocreatine, gamma, alpha, and beta peaks of ATP, and inorganic phosphate.
What's the field?
9.4 Tesla in this one. And as you probably need as much. Mouse heart is very small. It's like 100 milligrams. So even though it's nothing else there, it's still takes like 8 minutes to get the spectrum. And then if you stack them like this, you can see in time how this was stable heart, so it wasn't changing much at phosphocreatine ATP ratio over time. And all right, so I told you about the basic measurements. I told you about the free energy of ATP hydrolysis, and now a little bit about magnetization transfer. And I really don't want to teach you guys how this works, but usually I do show a little bit of how I understand this. All right, so you have an chemical exchange between two pools of phosphates. This is phosphocreatine and this is gamma ATP. And in the CK reaction, as I just told you before, there is a transfer between ATP and phosphocreatine. So how do we see the flux? Well, we irradiate the gamma with a specific frequency based on the chemical shift. And we continuously do this, not just like flip it and wait to come back as normal NMR, but you continuously suppress it, right? And as you suppress it, chemical exchange continues. And those which leave gamma go to phosphocreatine, start relaxing with T1 time constant, right? But before they do it, they are invisible in the phosphocreatine spectrum. So you have a decrease in the phosphocreatine peak from M0 to M infinity, which is like 4.8 seconds in our experiment. And then from this difference, you can calculate the flux. All right, so this looks like something like this. You irradiate gamma, it goes away. So this is the baseline. This is M0. This is M infinity. Gamma is gone. Phosphocreatine is smaller. And you can do the same for PI. It's just a little bit more tricky because PI is smaller. You need longer acquisitions because the noise, signal to noise will be not as good, right? But you can see that you can also measure ATP synthesis, transfer from phosphate from PI to gamma, because PI is also getting smaller here. And this is just an example of the work we've done. This is the Martinos paper I mentioned, published in NMR in Biomedicine, where we had the high-fat sucrose model. This is our mice, which are obese for 4 months on a bad diet, and the ATP synthesis in those was lower. And then it's important to mention, right? This is acute experiment. We take this heart, we hang them, and we add butyrate to the buffer, and it's just so amazing that the ATP synthesis increases this much. You know, these are the individual spectra for this, and delta G improves, right? This is the depressed delta G. We use absolute value. It should be negative, but we use absolute value, so it's positive number, right? So this is smaller at different parts of the experiment, where we first pace at the lower rate, 450 beats per minute, then 600 beats per minute, then we add high calcium with 600 beats per minute, double hit. And throughout the protocol, you can see that high-fat sucrose heart rates lower, and these are the controls, and the blue ones are the controls, and the black ones are the butyrate improved. So we improved delta G acutely with perfusions is butyrate, and here is just a plot of the delta G against the performance, and you can see the separation of the curves. It's really dramatic. And also I plotted circa threshold here. You can see that at the end of the protocol, these hearts were below the circa threshold, so circa couldn't work normally at this point. And this is just how it looks like in terms of acute experiment. You have the heart there beating, control diet, you add butyrate, nothing much happens. With high-fat sucrose diet, actually function increases. This is pretty amazing. It's actually inotrope. Energetic intervention increase the contractile function. Normally we use inotropes or now myotropes for that. And if we inhibited beta oxidation with bromocrictonic acid, the effect did not happen. All right, switching gears to C13, how we do it. So because C13 is such a weak, weakly it's not very visible by NMR, and it's only 1% of it in normal carbon. You have to enrich substrate with it and use long acquisitions to get some signal. But the principle is we perfuse the heart with differently labeled substrates. Either everything is labeled, nothing is labeled, or everything is labeled, or every other carbon can be labeled. And then from that, you get acetyl-CoA entering the TCA cycle, which can be either unlabeled, first one can be labeled, second can be labeled, or both, right? And then this goes through the TCA cycle, and it's a cycle, so it repeats several cycles and labels alpha-ketoglutarate in certain patterns. The issue is alpha-ketoglutarate concentration in the heart is low, right? Alpha-ketoglutarate, this is very low concentration, but luckily we have this malate aspartate shuttle exchanging alpha-ketoglutarate with glutamate, and then we can look at glutamate, which is in cytosol, and it's much more of it in the heart. So we do an extract of the heart and look at glutamate. Glutamate has 5 carbons, so it's 32 ways how to, and this can be labeled. So what we get is a composite spectrum of all these isotopomers, differently labeled, and we analyze that, feed it to the modeling software, and it spits out history of TCA cycle. How much of each substrate contributed to TCA cycle during the perfusion, which has to be pretty long to reach steady state, up to 90 minutes of perfusion. So this takes a long time. You first perfuse the heart for a long time, then you have to do the extract the right way, and then acquire 9.4 Tesla. It took us like 20 hours to get one spectrum. All right, and this is just an example how we use this approach. This is the paper I mentioned before, where we have hypertrophic hearts, hypertrophic cardiomyopathy hearts, and we measured. So glucose uptake is not C13. I didn't mention this when I was talking about phosphorus, but the way we do this is we use 2-digit glucose, 2-deoxyglucose, which doesn't get metabolized, but it gets trapped in the heart. So you perfuse the heart with 2-digit glucose, and then look at phosphorylated product, the first hexokinase phosphorylated glucose, and that accumulates in the heart. So over time, you can see the rate of glucose uptake. But and these others are calculated based on glucose oxidation, which is not on this slide, but we have fatty oxidation, we measure glucose oxidation, we calculate other things like uncoupled glycolysis, which means how much glucose came in and how much minus would get oxidized is your uncoupled glycolysis. That's how we call it. And then we also calculate other fates of glucose which didn't go to either one, didn't produce lactate or did not get oxidized. And all of this, you can see changes in the HCM group and normalized by EMPA. The protocol here was 4 months of empagliflozin treatment in developed HCM. And lactate production, so lactate production is from the effluent. You collect the effluent, the heart, you perfuse the heart, you collect the effluent, and you look at lactate in that. You can do C13 on the extract from the effluent, and then do C13 NMR on that. So we know how much lactate was produced during the perfusion time. And intracellular sodium, it was also elevated in the R403Q. This is the myosin model of HCM, and normalized by empagliflozin. Another thing that I want to mention, I published this only once. I never since then, because it's not that easy, and it's possible only in the hearts which have a lot of glycogen in them. But we perfuse with C13 labeled glucose and look at glycogen accumulation during the perfusion time. These are hearts which do have a lot of glycogen due to gamma-2 mutation, AMPK mutation. All right, and lastly, sodium and rubidium. So as I just told you before, maintaining low sodium level in the cardiomyocyte or any cell, especially excitable cell, is very important. Sodium should be high outside, low inside, to allow for all the excitation to be able to happen. But also, that's on plasma membrane, all those sodium channels. But then also in the mitochondria need low sodium environment to have normal calcium. If the sodium around them is too high, through this exchanger, they actually the mitochondrial calcium gets depleted. And again, that results in lower ATP production and higher ROS production. So this is an example I mentioned before. We had high-fat sucrose animals treated for 1 month with ertugliflozin, one of the SGLT2 inhibitors, and it did decrease the sodium and improve energetics and contractile function. You can see here control animals, high-fat sucrose animals, and here actually it seems like a little bit higher function in high-fat sucrose ertugliflozin treated hearts. Energetics was normalized, the diastolic function was normalized. Postocratic weight, we don't have delta G in this experiment, but PCR to ATP was lower in high-fat sucrose and improved by the treatment. And this is the sodium, much higher in high-fat sucrose group, decreased by ertugliflozin. And when we did acutely inhibit the exchanger in the mitochondria with CGP 37157, which is specific inhibitor of the mitochondrial sodium calcium exchanger, it actually did normalize PCR to ATP ratio in the acute experiment. So the idea there was if you normalize intracellular sodium, you normalize mitochondrial function and improve contractile function as well. Rubidium uptake, this is just I also just done one time in my lab. But what we can do, you can perfuse heart with rubidium, which is congener for potassium, and sodium potassium pump then pumps not just potassium but also rubidium. Then you can look at accumulation of the rubidium in the heart. And in high-fat sucrose hearts, the slopes tended to be lower, indicating lower NAK/TPase function in high-fat sucrose hearts compared to controls. All right, so and now switching gears again into other intervention we have. I mentioned before the skeletal muscle. We can increase skeletal muscle by either genetically by knocking out the receptors for myostatin signaling, or we can do a pharmacologic inhibitor of myostatin signaling in diet. No, actually, by injections, sub-Q injections. So the idea here is that, as I told you, larger muscle will act as a sink for glucose and BCAAs. This will increase insulin sensitivity. And as we all know, insulin is the major regulator of substrate metabolism in the whole body. And this should improve or normalize the substrate oxidation rates, fatty acid contribution to energy production, and decrease early hypertrophy and diastolic function. And this also is a work in progress. So this was an abstract from last year at AHA. As I told you before, high-fat sucrose diet increases the wall thickness. And when we had the mice, which had the light myosin Cree system-driven knockout of the myostatin receptors, the total wall thickness was lower. They had bigger muscle, 100% bigger pectoralis muscle, and 50 to 70% bigger all other muscles, and they improved the heart. Right, so nothing done to the heart, only to the skeletal muscle. Wall thickness was lower, diastolic function was improved. And when we did the same thing with the drug, and this is unpublished data, hopefully will be published soon. So again, this is intervention with antibody which inhibits the myostatin signaling in the skeletal muscle. And then we look at the heart, with the C13, the X-rays and all that stuff I mentioned. In high-fat sucrose, there is a much higher rate of glucose oxidation, lower rate of fatty acid oxidation, and the increased muscle fixes these changes. Also, the lactate production in the high-fat sucrose hearts is higher and improved by the skeletal muscle mass increase. All right, so I'm showing you this for the third time. You'll remember this is what we think is happening. But is it only heart which can benefit from this? Of course not. Other organs also suffer from insulin resistance. And it's known that insulin resistance and diabetes are connected to cognitive decline. And because this is a BrainMap seminar, I wanted to mention this. This is like a new branch of research here in my lab, because we have a new technician, excellent master student who is neuroscience masters, and she wants to look at this. So what we did so far, we think these mice are different in terms of behavior, but we only have a hint of that so far. We're doing another experiment where we're going to look at behavioral changes and memory formation in these mice. But when you just look at the activity, and when you have mice in metabolic cages, which we do for other reasons, you also measure activity. And then there are two axes you can look at, X and Y. X-axis cuts the cage along the short axis. So when you cross that, you go to eat or drink. But if you do Y-axis, you just go from one side to the other, we think it may reflect kind of curiosity, like they do it for just fun. And in x-axis, there was no difference in activity, but in y-axis, the high-fat sucrose mice don't do it as much. And the mice with bigger muscle do normalize this behavior. So that's just a new direction of our research. We want to look at brain because we have somebody who's interested in it, Sofia Mirioni. I think we are running out of time. So this is basically coming back to the heart. What we think we're doing. We changing metabolic milieu and switching the metabolic preference of the heart, which normally should be an omnivore, as I mentioned, and should be able to metabolize all the substrates. But in pathological conditions, it's forced to switch to mostly glucose oxidation. And also the entire furnace capacity of mitochondria, I call it, the ability to burn everything decreases. So as you increase fatty acid oxidation, you also increase the total ability of mitochondria to burn nutrients, including fatty acids, but including other things like branched-chain amino acids, if you've shown that. So by doing that, you improve the energetic charge of the heart, improve contractile function. But we now also know that it's reversing hypertrophy, which is a great thing. We just don't know how that is happening. So we have our work cut out for us to figure out exactly how this is happening. All right. So I just wanted to share with you, this is from 20 plus years ago when I was a postdoc. If you don't recognize me, this is me. And, and some people stuck with me, such as my wife. This is Jim Bolshoi, who did a lot of, basically in the old lab, this was NMR laboratory for physiological chemistry at Brigham. And this is Joanne Ingwall, who was basically pioneer of this. I'm saying this as I invented it, but no, the perfused heart. Joanne was one of the first who did this, perfused lung of hearts in the magnet and look at phosphorus NMR. And this is Rong Tien, who was my PI at the time. Again, Jim is still working with me. Currently, we're trying to develop at BU in vivo phosphorus, which is difficult in mice, but we're getting there. And this is Huamei, who is still doing with me the C13 stuff and extractions and all the acquisitions. And this is my current lab, or recent version of it. This is Tom. I mentioned a bunch of his work throughout the talk. And this is Sophia, which used to work here in Yifen's Yan lab, and she switched to us when she realized that she can get tissue remission if she works for me at BU. And this is Karen, which is right here, and she very kindly invited me to give this talk. Thank you very much.